Okay, hi everyone. Thank you for coming today. So this is our new-ish still seminar series that we're excited to get off the ground, highlighting early career researchers with preprints or recent publications. I'm Allison Barnes. I'm the vice chair of plant postdocs, and I'm currently a postdoc at NC State, where I study highland maize lipid metabolism. So today we have two great talks we're really excited about. And first is going to be Dr. Kevin Cope. So I'll introduce each speaker before their talk so that you don't forget sort of what their focus is. So Kevin is a molecular plant physiologist who earned a Bachelor of Science degree in plant science from Utah State University and a PhD in cellular and molecular biology from the University of Wisconsin-Madison. During his undergraduate studies, Kevin was awestruck when he learned about beneficial plant-microbe interactions in multiple plant science courses and decided to pursue doctoral research on a related topic. So in the NA lab, he received an NSF graduate research fellowship to study the molecular mechanisms that regulate ectomycorrhizal associations in woody plants. And after graduating, he shifted his focus to more agriculturally relevant symbioses and joined the Bucking and Supermanian labs at South Dakota State University, where he now studies both the arbuscular mycorrhizal and rhizobia legume symbioses using physiological, molecular, and transcriptomic approaches. And during his free time, Kevin enjoys spending time with his family and pursuing multiple hobbies, including gardening, biking, and chess. So thank you, Kevin. We're very excited to hear your talk today.
Thank you for the invitation. Let me pull it up. Is that showing up okay?
Yes.
Let me get a laser pointer here. All right, so yeah, like I said, thanks for the opportunity to come and talk to all of you today. It's great to have virtual resources like this where we can share research with one another after we've had such a long, isolated year. So I'm going to be talking about the research I've done on arbuscular mycorrhizal symbiosis in my current postdoc at South Dakota State University. And I'll be focusing on discussing the physiological and transcriptomic response of a legume species, Medicago truncatula, to colonization by both a high and a low benefit arbuscular mycorrhizal fungi. So I don't know how, I know one person on our list of attendees is very familiar with arbuscular mycorrhizal fungi, but I don't know how familiar others of you are. So I'll just briefly introduce what arbuscular mycorrhizal fungi are. So they're soil-dwelling fungi that belong to the phylum Mycorrhiza, subphylum Glomeromycotina, and they're obligate symbionts that associate with the roots of approximately 72% of plant species. So they're pretty widespread. Many of those species include agricultural plants that we use for crops. And they very likely played a key role in helping plants colonize land. If you didn't see it, a publication just came out in Science that an AM fungi made the cover. So this is a study on Marcantia, and it describes how that plant species potentially lured arbuscular mycorrhizal fungi into forming an association with AM fungi to be able to provide them with lipids. So anyways, it's a very interesting paper. I encourage you to go check it out and learn more about the evolution of the arbuscular mycorrhizal symbiosis. So here's a picture of what arbuscular mycorrhizal fungi look like. You've probably never seen them above ground because they don't form fruiting bodies like many other species of fungi do, like the Ceomycetes or Ascomycetes. But they form spores in the soil. And in the presence of roots, those spores will germinate. And these extra radical hyphae that you see around the root will go and come in contact with the root, form what's called a hyphopodium, and then they'll begin to grow intraradically. So inside of the root, if you use trypan blue, you can stain arbuscules or stain the fungi and see them present inside of plant cells. So this isn't very clear, but you can also use dyes like wheat germ agglutinin conjugated to Alexaphil 48, and you can see very clearly these intraradical hyphae passing in between cells and arbuscules forming in cortical cells within the root. So here's a close-up view of the arbuscules that form, and these are essentially a nutrient exchange site where mineral nutrients that the fungus mined from the soil are delivered across an interface with the plant cell, and then the plant in turn provides the fungus with access to carbon. So how do arbuscular mycorrhizal fungi help plants? I begin to allude to that, but they can essentially increase the ability of a plant to access mineral nutrients and water in the soil. This is partially because their extra-radical hyphae reach out into the soil much more effectively than roots can. They also can enhance plant tolerance to both abiotic and biotic stressors because of the additional access that they can provide to limited resources. And they can establish common mycorrhizal in plant. It sounds like someone's mic is not muted if you want to check yourselves. So here's a view of the arbuscule, just to help you understand how the fungus interfaces with the plant. So if this is a cortical cell, you can see here the fungus has penetrated into the cell, but the arbuscule that forms is surrounded by a periarbuscular membrane, which is a plant-based membrane that is enriched in nutrient transporters. Between the periarbuscular membrane and the plasma membrane of the fungus, you have a periarbuscular space, which is where the nutrient exchange occurs. So we can illustrate that with this diagram, where you can see the fungus out in the soil has access to phosphorus and nitrogen. It brings them to this interface. It exports them into the periarbuscular space, and then these phosphate and nitrogen atoms, for example, are taken up by the plant and assimilated into the plant tissues. The fungus benefits from associating with the plant because it provides it with carbon sources, including sugars and lipids, which are also exported into this interface and then taken up by the fungus and assimilated into its tissues for its metabolism. So arbuscular mycorrhizal fungi can provide a lot of benefits, but one thing that some people may not be aware of is that not all arbuscular mycorrhizal fungi are created equal. Some studies that were conducted about 10 years ago in my current postdoc lab, the Booking Lab, allowed for the identification of what are known as high and low benefit arbuscular mycorrhizal species. So they used a pretty clever setup where they used something called root organ cultures that are plant roots that grow in the absence of a shoot on a sucrose medium. And in these split plate systems that have 3 compartments, they allowed fungal hyphae to grow in media that had no phosphorus, and another compartment that had media with low or high phosphorus. And what they wanted to see is whether or not the plant would allocate 14 carbon to the fungal hyphae, and if there would be a preference for giving it to fungi that had access to phosphorus or not. And in the reverse, they put a fungal hyphae in a compartment with access to phosphate, and then had roots that did or did not have access to sucrose. I'm using this in some other studies. I'm briefly summarizing what they did, but they found that Rhizophagus irregularis, strain 09, has a lower carbon cost per phosphate transferred, per phosphate that it transfers to the plant, whereas Glomus aggregatum charges about 2.5 times as much. So pretty profound difference. So one of the questions that followed after that study, and that my current lab has been trying to understand better over the past decade, is whether or not a host plant can detect if an AM fungal partner is high or low benefit, and if so, can it respond accordingly and sanction the low benefit symbiont? So I'm going to partially address some of the research that we've done most recently by describing an experiment that is shown with this setup. So here you can see a plant that's growing in a root compartment and has been inoculated with arbuscular mycorrhizal fungi. And we used the 2 different strains, the low benefit strain, Glomus aggregatum 165, and the high benefit strain, Rhizophagus irregularis 09. In between the root compartment and the hyphal compartment is a barrier that has a 50 micrometer nylon mesh that only allows the arbuscular mycorrhizal fungi to grow into this compartment and excludes the roots from coming in, so that we can provide the fungus with access to potassium phosphate and 15 nitrogen ammonium chloride. Therefore, the fungus will take up these nutrients and deliver them to the plant. And this allows us to see, will the low benefit and the high benefit fungus differ in the amount of nutrients that they supply to the host? And if so, will the plant respond differently? So when we looked at root colonization rates, we evaluated the abundance of intraradical hyphae inside of the roots, and both fungi colonized the plant at about 90% root length colonization. When we looked at the abundance of arbuscules, it was about the same, no difference between the two. When we looked at the number of arbuscules, there was a significant decrease in number of vesicles for roots colonized by the high benefit fungus RAO9. And here you can see representative images of the colonization that occurred. When we then checked the growth response of the plant to these two different fungi, you can see that the only fungus that conferred a growth response was Rhizophagus regularis 09, the high benefit fungus. The other one didn't perform any better than the non-mycorrhizal control. This was likely due to the ability of the high benefit fungus to provide more phosphorus to the host than the low benefit fungus. And you can see here that although both of them did provide more than, provided more than was present in non-mycorrhizal plants, the high benefit fungus provided much more, both to the roots and the shoots. When we looked at 15N enrichment, we saw a similar pattern in the roots, but this pattern broke down in the shoots. So one thing to point out, when you're collecting root tissue, those roots are filled with intraradical hyphae. And so when you're measuring the amount of nutrients in the plant or in the roots, it's hard to distinguish between nutrients that are present in fungal tissue and nutrients that are present in root tissue. So an effective way to evaluate whether or not nutrients were transferred to the plant is by looking at nutrient contents in the shoots. And you can see here that although there's a slight enrichment of 15N in the roots of plants colonized by GA165 and RAO9, that increase does not occur in the shoots, except for the high benefit fungus. So it appears that, similar to what was observed previously, the high benefit fungus is providing more nutrients to the host plant. So just to briefly summarize that physiological data, although there were similar colonization rates, including the number of arbuscules that were present in the roots when colonized by either fungus, the high benefit fungus provided more phosphorus and nitrogen to the host plant. And the increased access to these limited micronutrients is probably what resulted in the positive mycorrhizal growth response induced by the high benefit fungus. And the elevated vesicle abundance and non-significant increase in 15N enrichment for the low benefit fungus and the roots that it colonized suggests that it's potentially hoarding resources and preventing the plant from getting access to them. So what's the plant response? What mechanisms are controlling these physiological responses? To evaluate that, we did an RNA sequencing experiment where we extracted RNA from both the roots and the shoots from 5 plants in each condition, including the non-mycorrhizal control. And after sequencing the data, sequencing the RNA libraries and analyzing them through a particular pipeline, we used DSeq2 to evaluate differential gene expression and a threshold of log2 fold change greater than or equal to 2 and a false discovery rate with a Q value less than or equal to 0.05 to determine differentially expressed genes. And so then we looked at the patterns of similarly regulated DEGs and we're able to do a cluster analysis, as you can see here on the left and the right, for root and shoots. And you can see that these clusters are sorted by gene expression patterns. So I'm going to first talk about, well, you'll notice that there's a lot more genes that are differentially expressed in the roots compared to shoots. And that's because the roots are what's being colonized by the fungus, so there will obviously be more changes that occur there. However, the shoot is attached to the roots, and therefore you're going to see some differences that occur, although not necessarily as dramatic. So you'll notice that within this gene expression cluster, there are genes that are upregulated by in roots that were colonized by either fungal species. But there's also some that are only upregulated by one species or the other, and genes that are downregulated in both compared to those that are upregulated in the roots. So we took this list of genes and we first did a targeted approach where we looked at genes that are known to play a role in the arbuscular mycorrhizal symbiosis. So for the past two or actually nearly 3 decades, there's been a lot of genes that have been functionally characterized and implicated as playing key roles in the arbuscular mycorrhizal symbiosis. And so there's a lot of genes that we could just check what their expression levels were, because we already know that they play an important role in the symbiosis. So I'll first describe some of my targeted analysis of those genes, and then I'll discuss a more blown out view of kind of a global analysis of genes that that haven't been described previously, but that may also play an important role. So one thing I didn't mention when I was describing the establishment of the arbuscular mycorrhizal symbiosis is that plants release a chemical known as stragolactone. That's a plant hormone that is a chemoattractant for arbuscular mycorrhizal fungi. It can stimulate the germination of arbuscular mycorrhizal spores and contribute to hyphal branching. And based on some recent work, it may also play a role in regulating how much colonization occurs based on feedback from cleave peptides produced in the roots that regulate arbuscular mycorrhizal colonization. And so when we looked at genes involved in that encode proteins that are involved in stragolactone biosynthesis, we observed that there were many genes that were more strongly expressed in roots colonized by the high benefit fungus. So to look at some of those, NSP1 and NSP2, CCD7 and MAX1A, you can see that in most cases, they're either more strongly upregulated in the high benefit roots colonized by the high benefit fungus, or exclusively upregulated in those roots. So because of that, it seems that the plant is enhancing stragolactone biosynthesis when it's colonized by the high benefit fungus to support colonization of the root by this fungus, but it may be striving to inhibit colonization by the other fungus based on the reduction in the expression of these key genes. So there's also been a lot of work done on identifying what's known as the common mycorrhizal or common symbiosis signaling pathway, which plays a role not only in the arbuscular mycorrhizal symbiosis, but also in the rhizobial legume symbiosis. And this pathway is shown in the middle here, involves the perception of what are called MYC factors, or more specifically, lipochitoligosaccharides. And following perception of these signals, there's a secondary messenger that transduces the signal to the nuclear membrane and activates various calcium channels that lead to differences in or fluctuations in calcium concentration in the nucleoplasm, known as calcium spiking. And these changes in calcium concentration are essential for the activation of a calcium and homogene-dependent protein kinase, CCAM-K, which is able to activate a transcription factor cyclops that then initiates the expression of many more transcription factors. And 2 of those, RAD1 and MIB1, were significantly upregulated in the roots of the high benefit fungus, more so than the low benefit fungus. When we looked at genes that we know are involved in nutrient transport, we saw that one that's involved in phosphate transport and another that's involved in ammonia transport were both more highly expressed in the high benefit fungus. And when we looked at a gene involved in lipid biosynthesis, specifically during mycorrhizal colonization, and a sweet transporter that's highly upregulated during arbuscular mycorrhizal colonization, these 2 were also more highly expressed in roots colonized by the high benefit fungus. So this kind of answers the question of how the plant responds to colonization with the high versus the low benefit fungus, and what's potentially driving the physiological responses we observed. More phosphorus and more ammonium are being transported to the host plant from the high benefit fungus, and in turn it's potentially receiving more lipids and sugars based on these gene expression patterns. So then we did this global analysis of DEGs to look at other mineral transporters, and some that came out that were particularly interesting, other than the ones that I've already described, ammonium and phosphate transporters, were a nitrate transporter, which it was rather ironic the day after, or the week after I found that this gene was so dramatically increased, a publication came out on rice and PNAS describing a nitrate transporter that was orthologous to this gene. So clearly it's playing a key role in nitrate transport based on these expression patterns and based on the functional analysis that occurred in that they performed in that paper. We also looked at sulfate transporters, which we didn't vary the amounts of sulfate provided in this experiment, but there was still significant differential expression of sulfate transporters in roots colonized by these different fungi, including sulfate transporter 1.1 and one that's annotated as MOT 1.1 and annotated as a sulfate transporter, but it's more likely a molybdenum transporter that may play an important role in this symbiosis. Beyond mineral transporters, we also look at secondary metabolite transporters and saw that there were some major intrinsic proteins that were strongly upregulated. NIP15 is a gene that's orthologous to NOD26, which plays an important role in the rhizobia-legume symbiosis and ammonium transport. And so it may also play a similar role in the arbuscular mycorrhizal symbiosis, but has yet to be functionally characterized. We also looked at additional sugar transporters because when sweet 1.2 was identified just 2 years ago, they found when they knocked it out that it had no effect on the symbiotic association. So they hypothesized that other sweet transporters might fill in and function in the absence of sweet 1.2. And so you can see here that sweet 3.3 and sweet 12 are both more highly expressed in roots colonized by Rhizophagus irregularis 09, but there are also others that are more highly expressed in roots colonized by the low benefit fungus. So we're curious whether or not these transporters may be expressed in different areas of the cell and might be sequestering sugars away from that low benefit fungus, but we're currently functionally characterizing each of these to determine their specific role in this symbiosis. So for sake of time, I'm going to skip the summary slide of what I just presented and just quickly show you some of the data from the shoots. So although there weren't many genes, we did see that there were consistently higher expression of genes involved in photosynthesis in shoots of plants colonized by the high benefit fungus compared to the non-mycorrhizal control. Some of those were also upregulated by the low benefit fungus, but they're consistently more strongly expressed in plants colonized by the high benefit fungus. So this may have contributed to the increased biomass that we observed in plants that were colonized by the high benefit fungus. If the genes involved in photosynthesis are more highly expressed, perhaps more photosynthesis is occurring, and therefore driving an increase in biomass. We also looked at marker genes for biotic stress response and saw that many that are upregulated during colonization of plants by pathogens, including fungal pathogens, some of those same genes were upregulated in plants colonized by the low benefit fungus. So you can see here, it's this gene, and this one as well are more highly expressed. Actually, all 5 of those are more highly expressed in the high benefit plants colonized by the low benefit fungus compared to the non-mycorrhizal control treatment. Similar story with the abiotic stress response genes. Non-mycorrhizal plants were very highly stressed because they were grown under low nutrient conditions, and the low benefit fungus only partially alleviated that stress because it wasn't transferring as many nutrients to the plant, whereas the high benefit fungus strongly alleviated those, and therefore we have a lower expression of some of these abiotic stress marker genes compared to the non-mycorrhizal control. So in summary, for the shoot response, the high benefit fungus increased biomass more, increased phosphorus and nitrogen uptake, drove photosynthesis more strongly, and only slightly increased defense responses, but strongly decreased abiotic stress responses compared to what the low benefit fungus did. And in the roots, similar story on biomass, phosphorus, and nitrogen content. But there's a huge difference in the number of I shouldn't say huge, but a significant difference in the number of transporters that were differentially expressed in roots colonized by this fungus as opposed to the low benefit. Much stronger differential expression over here. I didn't talk about defense responses and abiotic stresses in the roots, but if you want to learn more about those, you can go read the preprint. which carries the same title as the name of this presentation. And so let's finish by acknowledging the members of my lab, Heike Booking, my direct postdoc advisor, Arjun Kafle, a PhD student who is now a postdoc at NCSU, but originally performed this experiment, Kevin Garcia, who helped with the RNA-seq analysis, and Jaya Yaka, who's helping out with some follow-up experiments, as well as our collaborators and our funding sources. And of course, Plant Postdoc for inviting me to give this talk, and all of you for listening. So I'll take any questions.
So as a reminder, you can unmute yourself and ask your question or post it in the chat. I guess while everyone's thinking, I want to ask the first question, which I get to do because I'm the moderator. So you did all of this like really cool differential gene expression stuff. What did you like? Which one was the most surprising result to you? You know, because you talked a lot about like the targeted analysis, the things that you expected to see change. What stood out to you as being unexpected?
Some of the things we saw in the shoot response, I didn't necessarily expect some of those biotic stress marker genes to be upregulated, because that almost makes it seem like the plant's perceiving this symbiotic fungus as not a pathogen, but almost like a parasite. Mm-hmm. It's not parasitic because it didn't decrease the growth response, it just maintained it. But, you know, it's still there taking carbon resources from the plant. And so I guess it didn't necessarily surprise me, but I did not necessarily expect to see those genes upregulated.
I have a question that might be a little bit of a follow-up question to what you just said. Well, thanks for this very interesting talk. Speaking of the plant response in terms of the defense genes, I'm wondering if you were able to see the fungal gene expression in your data set, and if yes, if that gave you any insight.
I really want to do that. The problem is these aren't the sequenced sequenced species of arbuscular mycorrhizal fungi. And so I'm not sure how reliable the alignment of the RNA reads would be. However, I would like to potentially in the future collaborate with someone who has experience doing fungal transcriptome analysis and get help from them in knowing how to best align these. Because you could still align reads from a related species to the model Rhizophagus irregularis strain DOM 197198, the one that's sequenced, but I have not checked that yet.
But you have the reads, so in theory you could do that in the future.
Yeah, that's the goal.
Yeah, that would be really cool.
If they're not good enough quality, then we would probably we could repeat the experiment fairly easily and then extract the RNA from the fungus specifically with the goal of looking at the fungal response. But I don't have experience with that, so I would definitely be looking for collaborators to help with that.
It looks like you have a question in the chat, Kevin. Basically, I'll just summarize it by asking, what follow-up experiments are you planning to do to continue this story?
One thing I pointed out was a lot of genes that we could functionally characterize. So there's a lot of work that has gone into functionally characterizing nutrient transporters in their arbuscular mycorrhizal symbiosis. As I already mentioned, you know, ammonium, phosphate, nitrate transporters, but the sweet transporters. But this analysis was interesting because it allowed us to see very specific expression patterns of those previously functionally characterized genes. And so other transporters that haven't been functionally characterized that have a similar expression pattern, in my opinion, would be excellent targets for future functional analysis. And so, like I already mentioned, we're already working on the sweet transporters to further characterize the role that these different sweet transporters play when a plant is colonized by one fungus versus the other. but I would really be interested in following up on, like, I think it was NIP15, the sulfate transporters, etc. There's many more that I didn't have time to present today. So if I get an independent position soon, then I'll be able to start pursuing those.
Great.
Um,
Okay. Oh, sorry, go ahead.
Oh, sorry, I just had a question. Thanks for the interesting talk. I was curious, what time frame do you think it takes, how long do you think it takes the plants to decide whether a fungus is a high value or not? Because I thought it's interesting that the strigolactones gene expression has changed. Whereas if they're already thoroughly colonized, do you think that the strigolactones play a role in maintaining that colonization?
Yeah, one of the problems with this approach that we took is we just extracted RNA from the entire root system, which it'd be really interesting to do cell-specific RNA-seq, so you could see, or at least like regions of the root. The problem is you can't see where the fungus is colonized yet, so how do you isolate specific regions? So you're only looking at RNA from roots that are colonized versus those that are not. And so the approach we used was just a smorgasbord of all of that mixed together. So it'd be interesting to do both a time course study where you look at colonization at 2, 4, 6, 8 weeks, and then look at some of the expression of some of these key genes using qPCR instead of RNA-seq. It's so expensive. And that's something we attempted to do. Unfortunately, we got uneven colonization with one strain versus the other at different time points. And so that kind of messed with our gene expression data. And so our lab's currently moving to the University of Missouri. And so the goal is to follow up and do that time course experiment in the future to further clarify that exact question that you're asking.
Cool, thanks.
Okay, so I think now we will switch gears and we can continue all of this discussion after in our informal session. We'll move to our second speaker of today, which is Devang Mehta, who is a postdoctoral fellow with the URIG lab at the University of Alberta in Canada. So originally from India, Devang holds a master's in systems and synthetic biology from Imperial College London and completed his PhD in plant biotechnology with the Gruesome Lab at ETH Zurich in Switzerland. And he has a broad range of research interests and has published work on the limitations of CRISPR-Cas9 for engineering virus resistance in cassava, as well as invented a new technology for sequencing circular viruses and extrachromosomal circular DNA. His current research focuses on closing the protein gap in plant chronobiology using advanced proteomics techniques in Arabidopsis thaliana and its crop relatives. Apart from lab research, Devang writes popular science articles about genetic engineering and synthetic biology, as well as equity, diversity, and inclusion in the sciences, and has had his work published in magazines like Slate, Salon, and Nature. He is also a member of the Early Career Advisory Group at eLife and is a vocal advocate for reform in scholarly publishing. So welcome, and we are excited to see your presentation today.
Thank you, Alison. Let me just get my presentation started here. Do you see it?
Yes.
Okay, great. So, well, thank you, Alison and Sunil, for inviting me and organizing this series. I think it's such a great idea to highlight early career researchers, especially right now. So in today's talk, I'm going to be speaking about some recent work from our lab that's been focusing on using and testing different data acquisition approaches in bottom-up proteomics in order to address some technological limitations that have kind of limited our work in applying proteomics technologies in plant systems biology questions. So when when we're talking about systems biology, one of the starting points for this project was the fact that to do good systems biology, we really need good data, and by good data I mean data that is precise. So that has small error bars that has, it's not going to change depending on the instrument or the day that you're acquiring that data. Data that's reliable, that you're going to measure the same thing and it's reproducibly measured data. And then more importantly, especially for systems biology moving forwards, and especially for using it for engineering new types of crops, for example, we need to apply these, we need to have data that can be collected at scale. So the really important goal for systems biology, I think, is to have precise, reliable, and scalable data collection. And as a lab that uses proteomics techniques to study plant systems at across different genotypes as well as across different crops, and looking at complex experimentation workflows like circadian biology, which is what our lab focuses on, it was really important for us to kind of assess the different proteomics approaches that were available to us based on these metrics of precision, reproducibility, and scalable data acquisition. And if those of you who are kind of new to proteomics might not be aware of this, but modern proteomics methods are still not that great at measuring the entire proteome. So what I mean by this is, for example, if you compare it with the previous generation of gene expression measurement technologies like microarrays, for example, the latest microarray, which was released in 2008, so it's already more than 10 years old, was still able to profile about 48,000 features, covering pretty much the entire transcriptome of Arabidopsis. By contrast, for proteomics research, the very latest advances in proteomics technologies, this is an example, are still not able to get that depth of analysis. So we're still not able to acquire entire proteome scale data in single experiments using the latest proteomics technologies. So there's still a lot of work to be done in order to advance proteomics to a stage where we can actually have really deep data collection across an entire system. So then obviously the question arises, if proteomics is not that great, then why do we even bother using proteomics? And our lab's philosophy is that because proteins are so directly impacting a plant or an organism's physiology and metabolism, we really think that measuring protein levels it gives us a more direct indication of cellular state compared to, for example, measuring transcript levels. And previous work in our lab has found that, for example, if you're looking at the at differences in transcript levels across the time of day, which is across that is controlled by the circadian clock, you often find that protein levels are really badly correlated with transcript levels, mainly because of the fact that transcript levels are not indicative of are not directly linked to the eventual protein outcomes because of post-transcriptional regulation that can change the final outcomes even if you have measured all of your transcripts at the mRNA level. So that's really why we use proteomics technologies in our work. So just a brief introduction of mass spectrometry proteomics and what I mean by data acquisition in mass spectrometry proteomics, especially for people who haven't used the technology themselves. So typically for proteomics, you take your protein pool from your sample, you digest it using enzymes like trypsin to produce a population of peptides. And then you take those peptides and you inject them into your mass spectrometer. You ionize them and inject them into your mass spectrometer. And you first perform an MS1 analysis. So what we mean by an MS1 analysis is an analysis of the mass to charge ratio of each of these peptides that are injected in a mass spectrometer, and also gives you the intensity data that then correlates with the peptide abundance, which you can then use together with, you can collate that information for all of those peptides belonging to particular proteins to eventually get abundance metrics at the level of proteins. So this is at the MS1 level, but in order to actually identify what those peptides that you're measuring are and the sequence of those peptides, you need to do fragmentation of those peptides. So you select some of those peptides and you fragment them into smaller pieces, basically cutting off single amino acids at a time. And basically this allows you to then calculate the MS2 or to acquire MS2 data, which is basically giving you peptide sequence information by performing, again, M/Z data collection for each of these fragments. And then this allows you to calculate the peptide sequence, and you also get intensity data for each of those fragment ions that you profile at the MS2 level. So this is why this proteomics is often called MS-MS proteomics, because you have MS1 and MS2 data that's used for eventually coming up with protein abundance levels. Now, when you do this kind of a proteomics workflow, this bottom-up proteomics workflow, the two main decisions or two main parameter sets that you need to decide. The first set is, you need to decide what parameters are going to guide your MS1 level data acquisition. And this kind of, this first level decision impacts how many peptides can even be detected per chromatographic time at the MS1 level. The second decision is then, after the MS1 analysis has been complete, you then have to decide which of those peptides that you analyzed at the MS1 level do you actually select and take for fragmentation of MS2 analysis. So really together, these two decisions together make up the data acquisition approach used in a proteomics experiment. So the most conventional approach of doing mass spectrometry proteomics is something called data-dependent acquisition. It's also the more established and older workflow. And what typically happens in this kind of DDA acquisition mode in a mass spectrometer is that you perform an MS1 analysis, and each of these bands, for example, corresponds to a single peptide that's been identified at the MS1 level. And the darker the band, the more intense that peptide, the more abundant that peptide is. Now, typically in a DDA acquisition mode, you then select some of these peptides for fragmentation and MS2 analysis. And these MS2 analysis then produces individual spectra for each of those peptides that you have fragmented and measured. So what this means is that each of your MS2 scans corresponds and gives you sequence information corresponding to a single MS1 detected peptide. Now, a disadvantage with this approach is that it typically only measures the most intense peptides in a particular window. And this is also called a top N approach, which means that typically you're measuring about the top 10 to 15, depending on your settings, the top 10 to 15 most abundant peptides within that particular chromatographic window. Now, this obviously has a big disadvantage, which is that you're not going to be measuring this at the MS2 level, the lower abundant peptides in that pool, which means that you're going to bias your analysis towards measuring only the most abundant peptides in your specific chromatographic window. Another disadvantage with data-dependent acquisition is that if you do the same kind of experiment again, so if you repeated this experiment, suppose you had a technical replicate or biological replicate, and you ran the same sample again into the mass spectrometer, it's very likely that you don't have the same pool of peptides detected per chromatographic window, which means that you can have missing values in your data. So unlike an RNA-seq experiment where you have largely a complete data matrix where you're measuring every single transcript on all your replicates across all your samples, in a proteomics experiment using data-dependent acquisition, you often have missing values, which are then typically imputed or ignored or filtered out in your analysis. So this again reduces the depth of analysis of a proteomics experiment that uses data-dependent acquisition. So our lab has now switched over and is kind of benchmarking these data acquisition approaches, and we have now started working on data-independent acquisition approaches. Now, data-independent acquisition works a little bit differently. You perform the same kind of MS1 level scan where you measure all of your peptides at the MS1 level. But then instead of selecting only a few of those peptides for fragmentation, you actually take windows of peptides spanning defined mass to charge ranges. So you take all the peptides that lie in this window, you fragment them together to produce this kind of more complicated MS2 spectra. You do the same thing across the full range of your MS1 scan. So the advantage here is that you're actually measuring every single peptide at the MS2 level. You're not ignoring any peptide at the MS1 level. The disadvantage here is that the MS2 spectra is very complicated because in a typical DDA experiment, each of these spectra, you know that they correspond to a single peptide, so you can calculate the sequence of that single peptide. Whereas with the DIA approach, because these fragment spectra correspond to multiple peptides, you then need to perform a more complicated computational analysis. The more traditional approach is to use peptide libraries. So you have libraries of spectra that have previously been acquired corresponding to specific peptides, and you match those spectra with the spectra acquired in your DIA experiment to perform a library-based DIA analysis. However, more recent software developments have actually made something called direct DIA, which is a library-free data-independent acquisition method possible. So this is a huge technological advancement because it allows us to acquire DIA data without having these complex peptide libraries. So this is especially beneficial, for example, for non-model systems which don't have existing peptide libraries available. One thing that this kind of data independent acquisition approach does not address, however, is any issues at the MS1 level. So you're still performing a single MS1 scan. So you're limited to all of the peptides that are able to be detected at the MS1 level. But now we know that actually you often have peptides that are injected into your mass spectrometer that are not even analyzed at the MS1 level itself. So in order to solve that problem, we decided to come up with a method that we call boxcar DIA acquisition. So this is the third acquisition approach that we have been studying in this particular preprint. And what this method does is at the MS1 level, instead of performing a single MS1 scan, we perform two sequential MS1 scans, each of which only profile defined ranges of peptides. So they only analyze certain sets of peptide ions, and then the second scan analyzes the remaining set of ions. And then you perform an MS2 analysis, again, based on the data independent mode. So again, you're analyzing every single peptide that was acquired in this more complex MS1 type scan, this boxcar type MS1 scan. So this type of MS1 level analysis called boxcar proteomics was actually developed by the Matthias Mann lab at the Max Planck Institute in Munich. And all we've done in this study is combine it together with the data independent acquisition approach that's been developed by other labs. And what we, our hypothesis was that by combining these two approaches, we would be able to get better resolution at the MS1 level, which would then also be translated to better resolution at the MS2 level because we're using a DIA type MS2 acquisition. So in order to kind of understand, in order to understand how this kind of this more complex MS1 level analysis actually can result in benefits, you need to kind of look a little bit into detail at the way in which a mass spectrometer actually works. Sorry for this. Okay, so we need to actually just look a little bit into detail at how the architecture of a mass spectrometer actually looks like. So this is a kind of a cutout of a typical Orbitrap type mass spectrometer. So this is the mass analyzer in your mass spectrometer. This is the instrument that measures your mass to charge ratio of each of your peptides. And then you're injecting your peptide ions into this device through this thing called a C-trap. So this is a trap. This is an ion trap that basically traps all of your peptides together in that box, basically. And then these peptides are injected into your Orbitrap mass analyzer. Now, this is what a typical MS1 scan looks like. You can see that you have some very high abundant ions, and then you have some ions that are low abundant. Now, a limitation of conventional DDA type MS1 analysis or conventional MS1 analysis is that you can only flood this trap with a certain number of ions. So you can only fill that it has a limited capacity for storing ions. And what that means is that you typically have your high abundant ions kind of taking up space, and you don't have space left for lower abundant ions to occupy that area. So what you do with this boxcar method, and it's really a kind of paradigm shift in this MS1 level analysis, is that you kind of spread out your charges because you don't collect all of your ions together in that box. You actually distribute the ions and you collect only ions that belong to particular windows in your first scan. And then you perform a second scan that fills in the gap. And what you can see when you compare these two spectra is that you have a much more representative spectra in terms of the intensity. You don't have these high abundant ions kind of overrepresented in your data set. And it also allows you to kind of identify these lower abundant ions that might never have been analyzed in the mass analyzer, because there you have more of those ions accumulated in your trap over there. So this is basically kind of the more details about how Boscar works. But then this just brings me to our actual experiment, which was a benchmarking experiment to try and compare these different approaches to kind of develop a basic guide for people using mass spectrometry in plant systems, and to kind of give kind of a basic look at, what are the values, what are the advantages and disadvantages of these different methods for plant proteomics. So this was kind of our experimental setup. We used Arabidopsis cell cultures. We grew them in light and dark. We wanted a contrasting condition in order to do differential analysis. You then extract your proteins using a denaturing extraction. So this is simple STS extraction. You then digest your peptides. We use a robot called a Kingfisher device to do this. And then you inject them into the mass spectrometer. And we first compared the data-dependent acquisition approach that I described previously with the data-independent acquisition approach. And we used both a library-based DIA approach as well as a library-free DIA approach. And what you see quite clearly is that both of these approaches substantially outperform traditional DDA type analysis in the numbers of proteins that you can analyze, you can quantify. So, for example, in these Arabidopsis cell cultures, using a traditional DDA approach, we could only quantify about 4,700 proteins, whereas in contrast with DIA approaches, we had more than 30% increases in our depth of analysis. We then performed a subsequent experiment comparing this traditional DIA approach with our new Boscar data independent acquisition method. And again, we find a further improvement just by segmenting this MS1 scan into 2 scans. We're able to have an 8% increase in the number of protein groups that we identify. Now, one substantial advantage, so I mentioned before that a disadvantage of data-dependent acquisition is that you're only profiling the most abundant peptide ions at any given time. And this you can see actually in this data set where I've actually plotted all of the proteins that we detected based on their median intensity. So you can see that this is just a histogram of protein abundance in our data sets. And what you can see quite clearly is that you have a substantial pool of low abundant proteins that are not detected at all or not quantified in the data-dependent acquisition experiment, which are quantified in the data-independent acquisition experiment, which has a much more even distribution of peptides from low abundant to high abundant peptides. And you can see this even in more detail. So if you zoom into the lower abundant space and the high abundant space, for example, you can see that the data independent approach results in about 242 peptides over here at the lowest abundance, and you have 0 peptides at that abundance detected in the data dependent acquisition mode. So this is really just showing the advantage of DIA proteomics and offering a more even distribution of data, because it's analyzing all of your peptides across a wider range of protein abundance. The boxcar DIA method that we developed in our lab actually even builds up on this advantage of direct DIA by actually performing even better than direct DIA at the lowest abundant range, as well as identifying more proteins even at the higher abundant range. So it's better at both the lower abundance range as well as the higher abundance range. So I mentioned that a substantial disadvantage of data-dependent acquisition proteomics is that you cannot measure the same protein twice reliably across 2 replicate samples. So in order to assess that, we performed a subsequent experiment where we took 4 identical protein digests. So we took the same protein digest and we injected 4 different times into the mass spectrometer to do both data-dependent acquisition, direct DIA, so this is the library-free data-independent acquisition, as well as our new boxcar DIA approach. And what we found was that, so this is kind of a histogram of this data, and what you see is that the data-dependent acquisition approach, about 20% of the proteins identified using this approach were found in only 1 out of 4 technical replicates. So this is a huge problem for proteomics approaches, because if you're not able to measure the same protein across multiple replicates, then you really you're losing out on statistical power for differential analysis. In contrast, our direct DIA approach, as well as our new boxcar DIA approach, both were about about 99% of proteins profiled in both of these approaches were found in all 4 technical replicates. We also found that actually a lot of these missing values are due to the fact that data-dependent acquisition is really bad at profiling low abundant proteins. So when you look at these proteins that are found in either 1, 2, 3, or 4 replicates, and you plot them by abundance, you see that there is kind of a distribution where lower abundant proteins are less reproducibly measured in data-dependent acquisition. In contrast, for direct data independent acquisition, for library-free data independent acquisition, we find that most of the proteins are measured in all 4 replicates, but you still have a few proteins that are measured in 3 replicates out of 4 replicates. So you still have some missing values. In contrast, and this was really exciting for us, was that with boxcar DIA approaches, you have absolutely 0 missing values. And this was found both in an Arabidopsis experiment, which is the data I've displayed over here, where we repeated this experiment using HeLa cell extracts from human cells, and we see the exact same result where we have no missing values in the boxcar DIA data set. So I mentioned that both boxcar DIA and direct DIA were much better at identifying and quantifying lower abundant proteins. So we were kind of curious about, does this have any functional impact on proteomic studies that are looking at, for example, differentially expressed genes or differentially abundant proteins across different samples? And what we found was that if you plot the proteome of Arabidopsis based on abundance, so this is a plot showing every single protein in Arabidopsis plotted based on its abundance rank. What you do see is that there is this kind of grouping by function, where proteins related to glycolysis and translation, for example, are very high abundant overall, whereas proteins related to proteolysis and carbohydrate metabolism are kind of at the median abundance range, whereas important regulatory proteins like protein phosphorylation-related proteins like kinases and phosphatases or transcription factors, these regulatory proteins are typically present in lower abundant, at lower abundance within cells. So this is really important, because if you're using a data-dependent approach for your proteomics experiments, you're often probably missing out on identifying as many of these important regulatory proteins as you could if you use boxcar DIA and direct DIA. And what we found was that direct DIA is better at identifying protein, or does identify more protein phosphorylation-related proteins, as well as transcription factors in Arabidopsis cells. And boxcar DIA is even better than direct DIA at quantifying these lower abundant but extremely important protein groups. So I mentioned that we did these experiments, we did these benchmarking experiments in both light and dark grown Arabidopsis cells. And the reason we did this was that this cell culture, Arabidopsis cell cultures, are a really widely used platform for interactome experiments. So for example, affinity purification experiments, turbo ID experiments, biotin labeling experiments often use Arabidopsis cell cultures, but we've seen that a lot of the literature often uses light or dark grown cells kind of interchangeably. And in this study, we just wanted to show that there is a huge difference in the protein complement that can be detected by the mass spectrometer in light and dark grown cells. And what we find is that there are more than a third of the proteins that we quantified in these different approaches are differentially abundant between light and dark grown cells, which is kind of obvious. But we then were able to give a list of these proteins. So then, for example, if research groups are interested in performing interactomics experiments using proteins involved in, for example, protein transport, then you might want to use dark grown cells, because those proteins are more abundant in dark grown cells compared to light grown cells. By contrast, light grown cells are obviously better for performing interactome experiments with photosynthesis, chloroplast translation, carbon fixation related proteins, for example. So this was just kind of the resource that we presented at the end of this paper. So just to summarize our main results, we found that this conventional DDA proteomics approaches have serious disadvantages that impede its use, particularly with complex experimental designs like time course experimentation, which is what I'm interested in as a circadian biologist. And then we also found that boxcar DIA, which is this new approach that we developed combining boxcar mass spectrometry with DIA type MS2 acquisition, offers 100% reproducible quantification of the same set of proteins between technical replicates. And this, thus solves this long-standing missing value problem in quantitative proteomics, especially label-free quantitative proteomics. Next, we found that boxcar DIA does offer better quantification of lower abundant protein classes that are nevertheless extremely important in terms of their regulatory function, like kinases and transcription factors. And then another thing I should mention here is that all of these gains in boxcar DIA and direct DIA were realized with no increase in cost or analysis time compared to the conventional DDA approach that we used in our lab. And I guess the kind of key message from our paper is that we really think that researchers using quantitative proteomics in integrated systems biology in plant systems, for example, should definitely start adopting newer methods like boxcar DIA or other data-independent acquisition approaches. In my own work, this is a really beneficial improvement because I told you I'm interested in performing circadian time course experimentation, and this involves performing, taking samples at multiple times a day from the same plants or from the same plant population in order to track the dynamics of the same protein across time. And this can become a huge problem if you're using data-dependent acquisition, because if you are missing values at different time points for the same sample, then you are not able to get this protein level dynamics. And this is actually a recognized issue in circadian biology right now, where recent research, including by my own PI, has found that if you look at the transcriptome, about 30% of the transcriptome is dynamically changing in response to circadian regulation. But if you measure the protein level changes across the time course, you actually don't see much protein level changes. And one reason that we think that previous experiments have not been able to see protein level dynamics changing across the circadian time course is because of the fact that you had a lot of missing values, which resulted in an absence of complete quantification of the full protein dynamic trajectory across the day. So with that, I'd just like to conclude by thanking my supervisor, Dan Ulrich, whose lab I've joined, Dr. Sabine Scandola, who's a fellow postdoctoral in the lab, who has helped with this research. We also had some technical support, especially in terms of software that we used by Biognosis AG, which is a Swiss company that develops software that's able to deal with data-independent acquisition approaches, and then funding by the Canadian Foundation for Innovation, as well as the National Sciences and Engineering Research Council in Canada. And with that, I'm really happy to take any questions.
You have any questions? Okay, I can ask the first one. So I have done some mass spec, but I'm not like a mass spec expert. So I'm not as sure like what exactly all the setting differences are. But do you think that these sort of pipelines that you're developing for proteomics could eventually also be applied to like untargeted metabolomics and other quantifying other compounds via mass spec?
I have never done metabolomics experiments, so I don't know if I can really speak to that, but I don't think they would be directly applicable because I guess you're not performing this kind of MS2 analysis, right? You don't have, I guess you're not fragmenting ions and then analyzing them in metabolomics. But I might be wrong. I'm completely new to metabolomics.
Okay, sure, yeah, that's no problem.
Yeah.
Thank you for your presentation. I really enjoyed that. as someone who knows almost nothing about proteomics, that was very helpful for helping me understand it. Well, I shouldn't say understand it now, but have a better understanding of it. So for someone like me who's a novice with proteomics, what recommendations would you have for designing an experiment, perhaps as like a follow-up to the one that I presented on, to look at protein levels? Because in my presentation, I talked about expression of genes, but we know that gene expression doesn't necessarily mean protein translation and therefore activity of the protein. So it'd be great in the future for me to do experiments where I not only look at the transcriptome, but also the proteome. What are some key things you'd say to someone like me to ensure that I can do both RNA extraction and protein extraction with my samples and get reliable results that could complement one another?
Yeah, so I mean, that's a really good question. It's something my lab also does, is doing RNA-seq as well as proteomics. And one, I guess the kind of the main thing is the sample requirements are quite different. So for example, you obviously can't amplify proteins, so you need to start with a lot of starting material, especially if you want to do things like phosphoproteomics, if you're trying to measure changes in post-translational modifications. So if you want to get some more regulatory information, you need to have a lot more starting material. And then in terms of experimental design, one thing I did not touch on in my talk is using labels like tandem mass tags. I don't know if you've heard of these, but there is another approach of proteomics called labeled, using chemical labels for quantification. And that is a method that I would recommend if you have a very small set of samples, because those labeled experiments can typically only do about 16 to 18 samples. That includes your replicates. But for more complicated experiments, for example, if I'm doing a time course experiment with multiple genotypes and multiple replicates, then I would definitely suggest that you use one of the methods that we described in this paper, either the direct DIA or the boxcar DIA approach. Yeah, and then I'm obviously happy to answer more detailed questions over email, and our methods are publicly available on bioRxiv. But yeah, the main consideration is both the sample amount. You'd need a lot more sample just in general to extract your proteins. And then the second is, how many variables are you measuring in your experiment?
As a follow-up to that, is proteomics as expensive as transcriptomics?
Oh, that's a good question. It kind of depends on what resources are available to you. So we have our own mass spectrometer, so we don't need to pay for time on the instrument. But some facilities I know do charge like by time, so that becomes a little bit more challenging, because each sample in our experimental approach, for example, took about 2 hours to acquire. So you can imagine that you need about a week's worth of time on your mass spectrometer instrument to get that. If you're paying for those hours, then it's quite expensive. But if you have a collaborator or someone who's willing to do those samples without charging you for the time, but just charging you for like sample preparation, then it's much cheaper than RNA-seq.
Thank you.
So for reference, I can say that I think at our lab, it costs about $75 to $100 per sample.
Any other questions before we transition over to our informal group chat? Looks like no. So, okay, thank you so much for your talk. It was great. It's kind of fun to see, to get into the more technical side of things sometimes and learn about new techniques and stuff like that.